Hello, and welcome to Pint of Science, the world's largest science festival. I'm your host, Andrew, and this week we are in Cambridge, a city famous for its pub. But because of the current pandemic, we are bringing you the show live to you in the comfort of your own homes. On tonight's show, we'll be focusing on the immune system, how it fights disease, how it can cause disease, how it's affected by your poo, and what's it got to do with pregnancy. In addition, we're privileged to give you a sneak peek at pieces of artwork created by talented local artists working alongside our speakers in our collaborative project, Creative Reactions. You can find more about this in the description below, and we'll be posting links throughout the show. Right, speaking of the show, this is being streamed live on YouTube and Facebook. So hello to all our fans out there. This show is made for you, so if you have any questions for our speakers, leave a comment. If you're watching this as a replay, give us a like and hit that subscribe button. Right, as a scientist, I find the immune system absolutely fascinating. How it's there working all the time, protecting us from pathogens, disease-causing microbes. But what happens when a new virus comes along and the immune system, how does it react? How do we react? Dr. Harriet Groom is a researcher from the Department of Medicine, University of Cambridge. She's also a fellow of Sydney Sussex College, and she studies the interactions between viruses and human cells. Harriet, lovely to have you on the show.
Lovely to be here. Hey. So no one needs convincing at the moment that viruses are important. Even in the pre-SARS-CoV-2 times, who out there listening has not had a viral infection? Nobody. So to stop viruses, we need to understand them. And in this little peek into cellular immunity, I'm going to use HIV, the virus that causes AIDS, and SARS-CoV-2, the cause of our current COVID-19 pandemic, to get you to think about how cells and viruses interact. And I'm going to use two examples of how our body have evolved to limit HIV, and at the end touch on some work that some people are doing in Cambridge with SARS-CoV-2 to inform future therapies. So time for my hard sell on virology that hopefully no one needs. So what is a virus? So I've taken this actually from BBC Bitesize. They consist of a strand of nucleic acid, so either DNA, which you'll be familiar with, or RNA. And this is surrounded by a protective protein coat, a capsid, and sometimes by a membrane too. And they replicate inside cells and so are obligate intracellular parasites. So why study viruses at all? Well, firstly, viruses are everywhere and in huge numbers. And from a human-centric point of view, I mean, as we are all acutely aware, they cause disease. But not just in us, also in things we care about, like chickens or tobacco. And for reasons we'll come on to, viruses take over cellular processes, so they can tell us a lot about how things work and how they're controlled. So, for example, this is a picture of some cells in cervical cancer, and 99% of cervical cancer cases are caused by a virus, the human papillomavirus. And viruses can be repurposed medically, for example, as vaccines or as experimental tools, not just turning mice green. They can also be used for things like gene editing, which you will have heard quite a lot about recently. Now, I don't know if any of our viewers have come across the website XKCD, but it has a section for answering absurd hypothetical questions. And one of them is, what if every virus in the world was collected into one area? So how many viruses do we reckon are in a typical healthy human body? Well, the very approximate answer is 3 × 10⁻², which is a pretty big number. And if you multiply that by the roughly 7 billion people alive in the world at the moment, then you get enough virus to fill 10 oil drums, which doesn't seem like very much, but viruses are very small. So then if you start to think about all the organisms on the planet, this brings the number up to 10⁻31. Now that really is a hard number to imagine. So let me help. If we took all the viruses in the world, according to these calculations, and piled them up, that would make a small mountain. And if we lay them end to end, they would spread for 10 million light years. And the next time you're killing time, type 10 million light years into YouTube, and you'll find this amazing video which zooms you out 10 million light years and back again from Earth, and you'll really appreciate how far that is and how many viruses we have on the planet. So everyone will have become very familiar with the concept of a viral outbreak. But viral outbreaks are happening all the time in the human population. So this is a screenshot from a website called Health Map, showing incidences of reported outbreaks just in the past week. So some of this is obviously SARS-CoV-2, but by no means all. So there are outbreaks happening all the time. And some of those from earlier will be familiar to you. So you may remember Zika virus, which is famed for its link to microcephaly, and Ebola, which is this example here, and now, of course, SARS-CoV-2. Now, in the next few minutes, I'm just going to try and convince you that we need to understand how viruses interact with cells in our bodies, and that knowing this can help us understand normal cell functions and how things can go wrong. So this brings me to HIV, which is the virus that I've worked on for the past 15 years or so. And HIV is human immunodeficiency virus. It's a causative agent for acquired immune deficiency syndrome, or AIDS. And some people in not very affected countries feel like this is a kind of solved problem, but it's not. So there are still 37 million people across the world that are infected with HIV, and there are roughly 1 million new infections every year and 1 million deaths. So it causes immune deficiency by infecting cells of our immune system called T cells, and these are a vital part of the immune system. And with their declining numbers, then people become more susceptible to infections and also cancer if it's not treated. Now, retroviruses are so called not because they have a poor dress sense, but due to an ability to reverse transcribe their genome. And I'll just explain what that is. So this is a simplified version of the life cycle of a retrovirus. So this is the cell, and this is the nucleus inside. And the HIV will bind to the cell surface, and that fuses with the membrane on the outside of the cell, and it empties its contents inside. And the capsid that I mentioned at the beginning releases the genome into the cytoplasm, which is the RNA genome. And then that is converted into DNA. Via this process of reverse transcription. And this can then be taken into the nucleus, where it can enter the host cell DNA in a process called integration. And this is a hallmark of another hallmark of retroviruses. And it's really important because it allows the virus to make lots of copies of itself, to evade the immune system. And it can also be important because it can disrupt genes leading to cancer as well. Now, we have anti-HIV drugs that work by targeting different steps in this pathway shown by the little pills here. But HIV can evolve to develop resistance to these drugs. And this is because viruses are in a constant battle with the immune system, and so have evolved to be the ultimate cell biologists. So evolutionary selection alters their appearance and replication methods to avoid the immune system. And one way to fight back is to look at molecules in the cell that the virus interacts with. And these aren't encoded by the virus, and so don't evolve resistance in the same way. And I'm interested in a subset of the proteins in human cells that stop viruses like HIV growing, and these are called restriction factors. I'm going to talk very briefly about two of these. So firstly, number one is SAMHG1. Which stops HIV growing in certain immune cells by breaking down the precursor of DNA, DNTPs, shown by the little lightning here. And this stops the virus genome being made in that process of reverse transcription. This role also makes it really important for other things for the cell, and it's been linked to lots of things, including cell metabolism and DNA repair and regulation, and so also cancer. And so lots of people are thinking about SAMHG1 as a therapeutic target, and I want to know what happens to cells if you try and alter your levels of SAMHG1 in the cell. I'm also interested in what the general variation in this protein is among the healthy population. Now, the second thing I'm interested in comes back to this. So I said that 37 million people in the world are infected by HIV, and you may know that the vast majority, so 70% of these, are found in sub-Saharan Africa. However, despite this, only 3% of genetic studies until recently have considered African sequences. So thinking about African DNA in African people. And my collaborators have recently identified an African-specific genetic variant so which is represented by this, which is strongly associated with a reduction in the level of virus in HIV-infected people. So together we've shown that there's another cellular protein which is responsible for this. But the big question we're looking at the moment is how that happens. And then lastly, in these uncertain times, my expertise, like for many virologists at the moment, has been redeployed against pandemic SARS-CoV-2. And I'm using some of my knowledge from my PhD about viral RNA to work with lots of groups in Cambridge to look at the shape and role of RNA structure in infection. So I've shown a piece of the SARS-CoV-2 genome here, which is made of RNA, and RNA can form these elaborate structures shown here, which are important throughout the life cycle, but are quite poorly characterized. So we're looking at how these structures form, how they change, and how they interact with themselves and other things in the cell. And so they can be quite important therapeutic targets and can also inform vaccine design as well. So if you want to know more, then please have a look at my website or follow me on Twitter. I'm a research fellow at Sydney Sussex, as Andrew said, and these are the lovely people that have funded the work that I've talked to you about today. And thank you very much for listening.
Thank you very much, Harriet. That's really interesting. I didn't really know about, there's so much stuff we just don't know about SARS-CoV-2 and the structures of the RNA. You know, at school, I learned that RNA is just long strand of RNA. But yeah, it makes these elaborate structures. Right, before we move on to comments and questions, have you seen the artwork created by Creative Reactions artists based on your work?
Yes, it's amazing, especially the speed with which it was created is absolutely fantastic, and it was real privilege working with Naomi.
Yeah, look at that. So this was done in watercolor or something?
Yes, it's really so I just showed her a picture of a molecule that I'd used for an exhibition in the university library to show how SAM-HG1 is breaks down DNTPs, and it's transformed into that, which is just fantastic.
That's amazing. Right. If anyone wants to know more about the artwork, we've got a link that we shall share in the description below. There it is. And yes, please do go and have a look at that website. Right. Shall we have a look at questions and comments? Do we have? Yes, we do. We have one from Marcos. Is reverse transcription prone to errors, and could that cause mutations in viruses?
Wow, that's a brilliant, really interesting question. And yeah, mutation of viruses is something that's very much on our minds at the moment. And one thing about HIV is it's very prone to accumulation of mutations, which is why resistance is really important. But one of the reasons for that is not so much the reverse transcription process, but the fact that it has an RNA genome, and RNA is much less stable than DNA. And it's one of the reasons that if we originated in an RNA world, one of the reasons we might have transitioned to a DNA world to have this much more stable, less prone to error genetic makeup.
Right. Okay. So we've got a second question from Kate Grant. Did you have any surprises when you did your research on SARS-CoV-2?
Well, yeah, I mean, I'm certainly evolving. It's a new area for me. My first coronavirus project, and I think we're all learning a lot every single day. The number of papers coming out, and I listened to This Week in Virology, which I'm going to name check. It's an amazing podcast that has lots of up-to-date and well-balanced and informed discussion about SARS-CoV-2 and in normal life other viruses. So I'd recommend that.
Excellent. Do we have another comment? Oh, round of applause. Yay.
Oh, thank you. To you too. Thanks for tuning in.
Quick thing. So you mentioned chickens in the beginning. So apparently I've got a quick fact about chickens in that one of the first coronaviruses to be identified and described were found in chickens. And that was called infectious bronchitis virus. Lovely name. Yes,
yeah, one of my friends works on that. Sneezing chickens is a very interesting thing to hear.
Excellent. Thank you very much. Right, so thank you very much, Harriet. Should we move on to our next speaker now? Okay, right. So I don't know about you, but I love cakes and I love biscuits. But for many people, the gluten in these types of food can cause a debilitating condition called celiac disease. This is where the immune system starts attacking the gut. Dr. Elizabeth Souilleux is a senior lecturer and honorary consultant at pathology at the University of Cambridge. She's also the Director of Studies at Churchill College and an associate professor at the University of Oxford. Liz, it's lovely to have you on the show, and over to you.
Great, thank you very much, Andrew. So yes, Andrew's mentioned some of the things I do. I think maybe it's worth just mentioning that I'm a pathologist as well as a researcher. So a part of my world is about making diagnosis. And we make diagnoses on my team for lymphomas, which are tumours of the immune system. This is nothing to do with lymphomas. This is repurposing some of my knowledge from lymphomas in a completely different way. And it starts with a rather personal journey, which I shall tell you a little bit about to explain how I had the idea. So I had two children. My old one is 9 now. Ben, bottom left there, after about 2 years, became very anemic and really not very well, and had terrible tummy pain and many of the other symptoms that you can see on this slide. Lydia was much more sick, didn't sleep, vomited, and was a complete nightmare in short. You can see her here eating a pasty, which was a disaster, as you can imagine, because it's full of gluten, but I didn't know at the time. Eventually, I worked out that my children probably were celiac and needed a gluten-free diet. By the time I'd taken them off gluten, it was really not possible to test them anymore because people need to eat gluten in order to be tested for celiac disease. And we know that although people like myself, pathologists, do look at biopsies under the microscope to diagnose celiac disease, and that is the gold standard, which I'll show you in a moment, that actually it may not be the most sensitive of tests. And there may be up to 2% of the UK population who are out there without diagnosed celiac disease. So we probably need a better test. This is just a sneak preview of some of the artwork. I've been a bit naughty putting it in. This is Kate Grant's resin of some duodenal or small intestinal biopsies. I think they're wonderful. I'm going to disappoint you now and show you what they really look like in real life. So these are the type of samples that are regarded as the gold standard for making the diagnosis of celiac disease. Up on the top right here, we have normal duodenum, and the tissue has these finger-like processes or villi that many people will have learned about at school that stick out into the gut to increase surface area. Meanwhile, when you've got celiac disease, those villi get all short and fat and don't increase the surface area properly, meaning you can't absorb food properly. And the lymphocytes, which you can see here as little purple dots in normal, increase in number quite substantially because these lymphocytes, which are cells of the immune system, are unfortunately the bad guys in celiac disease. This looks simple. It is not. Actually, looking at small intestinal biopsies under the microscope is a bit like looking at oak trees. They're all subtly different, but actually quite similar. So if we said that half of these were the equivalent of celiac disease and half of these oak trees were the equivalent of normal, can you divide them into two separate groups? And I think most of you would agree you couldn't. So actually, trying to make the diagnosis is quite challenging. So I thought hard about, is there a way we can make a better test? Obviously, you want a reproducible test, and it needs to be objective, ideally not depending which pathologist looks at your biopsy and what their threshold for calling something celiac disease might be. If possible, you wouldn't want to have a biopsy at all, because then you could avoid having an endoscopy to get that biopsy. And you probably wouldn't want to eat gluten at all because it makes you ill. So bearing all those facts in mind, and the fact that my whole world as a lymphoma pathologist revolves around lymphocytes, which include T cells, I thought, well, maybe we should be looking at these T cells in more detail because they're actually the problem here. Let's use them for the test. So let's think a little bit more about T cells. Again, they're a type of lymphocyte, as I said. They're a type of white blood cell, so they're part of your immune system. But in celiac disease, they not only respond to gluten, but in the presence of gluten, they attack the gut, which is not very helpful. Here we've got a depiction of all the chromosomes in the body. We have 2 of each number up to 23. And for a T cell, it has a receptor, which is the thing that binds whatever it can get hold of in order to attack or respond to. And that receptor has 2 proteins that make it up. So those are encoded by 2 separate genes within our genome. And for gamma delta T cells, which are quite a rare type of T cell, they're on chromosomes 7 and 14. And the really amazing thing about the DNA in these T cells is when the T cells are growing up, when they're developing, they rearrange their DNA in a way that no other cell type does. And that gives each of these cells a unique barcode, which is the bit of DNA encoding the receptor, the thing that determines what it can bind. So various groups have thought hard about T cells as a way of looking for a diagnosis of celiac disease. And they've said, well, if we happen to determine that specific barcode, that specific DNA sequence that is relevant to celiac disease, well, if we looked at all of the T cells, and each T cell here is depicted by a circle, in a biopsy, could we just find a few of those relevant sequences and say that person has celiac disease? But what if unaffected people have a smaller number of them? And what if you don't identify these? It's a bit like looking for a needle in a proverbial haystack. So we made a slightly different model where we thought, actually, let's think about it a different way. Let's assume that all the T cells in somebody with disease are a bit different to somebody who doesn't have the disease, except that there are some that overlap between the two, rather than saying we have to look for individual sequences, just as specific T cells that will tell us the answer. Let's look at all of them at once and see if we can say, by looking at them all, can we determine who's got celiac disease and who hasn't? So we took some biopsies of small intestine or duodenum, and we took the DNA out of those biopsies and sequenced all of those T cell receptors, all of those barcode sequences that I've told you about. We used some complex maths, which we developed ourselves, and we've been lucky enough to file a patent application for because it's really quite novel stuff. And then we used our mathematical analysis to work out which samples were more like each other and which were more distinct from each other in order to see if we could make the diagnosis. So here we've got a graph, or not a graph, it's a cluster plot that shows you the samples. Each of these numbers is a different patient. Patients who have celiac disease are in blue, and those who do not are in red. And what we've done is clustered them together in such a way that the longer line you have between them on this axis, the more different they are from each other. And so what you can see is, using our method, looking at all those T cell barcodes in a sample, you can cluster together pretty much all of the celiac patients together, relatively separate from the normal patients. There are a few normals that might be in the wrong place, but who knows, they may have undiagnosed celiac disease. We don't know that. So we then thought we'd better validate this. Let's see if it really works. We've got some more samples from other celiac patients. These are all very abnormal biopsies without those long finger-like villi I told you about. And we took the DNA from those, put it through our method, and lo and behold, you can see them here in black. They've clustered into the right area on our plot. They are like celiacs rather than like normal samples. All well and good, but can we take this further? So the next thing we did was considered those same patients we just used after they'd had at least 6 months on a gluten-free diet. So the question we're really asking is, if we got people who are not eating gluten, can we get the diagnosis right using our method? And here you can see again, all 4 of those patients have clustered closely with the celiac samples and not with the normal samples. All the other tests we do for celiac disease would get these patients wrong, particularly the histology looking down the microscope. We would say these look normal, but they are actually not normal because the lymphocytes in them are the celiac-causing lymphocytes, and it's those that we've been able to detect. Finally, we've had a look at whether we could actually do this analysis on lymphocytes, on white blood cells circulating in your blood, rather than having to do a biopsy. And you can see that the celiac samples in blue here are sort of clustering together. It isn't perfect, but I think with some small changes to our mathematical approach, we might be able to do this on blood and avoid an endoscopy altogether, but this is all very much a work in progress. So just to summarize, I've told you about T cells that are a type of lymphocyte, they're a type of white blood cell. And I've told you how unique parts of their DNA, which have been rearranged as they developed, act as barcodes, but they also encode these receptors, determining what they can bind. And by sequencing those receptors with some clever maths to analyze the sequences, we can group together patients with celiac disease and separate them from unaffected people. This approach even works if the patient isn't eating gluten, which is potentially a major step forward. We don't know whether this would work adequately as a blood test yet, but we're certainly going to have a good go at getting it to work so that patients can avoid an endoscopy. The work was funded by Coeliac UK and a variety of other organisations, including Innovate UK, to whom we're extremely grateful. And finally, I'll leave you with a summary. Thank you ever so much for listening.
Thank you very much, Liz. That is really, really interesting. It is a game changer, isn't it, to actually diagnose people who don't even know if they've got celiac disease.
I hope so. I really hope so. And I think there may be a lot of people out there who actually don't realise that's why they don't feel very well. And there are others who've been frustrated because they just haven't been able to get a test. But I think by applying our understanding of the immune system, actually, we can think of better ways to make the diagnosis like this one.
Right, yeah. Right, I think we've got a few comments coming in. Do we have... Yes, we do. Right, so we've got one from Graham. As it works, if the person is not eaten gluten, does it work if they never had?
That's a really interesting question. I think if you've never eaten gluten, you probably wouldn't have celiac disease because you've got to be exposed to the gluten in order to make a response to it in the first place. So while you might have the potential to develop it, if you've never had any gluten in your life, and there have been populations in the world traditionally, particularly parts of South America and places where gluten just hasn't been eaten, the Eskimo population have probably never had any either, then maybe they have the potential to develop it, but they actually don't develop it. But it's not something that's easy for us to test because we don't have access to people, to our knowledge, who've never eaten gluten.
Yeah, so as an Asian myself, we eat gluten as 100% gluten in some of our products. And so I've never really heard of anyone, any relatives who've had celiac disease. So it would be really interesting to see if there's any kind of geographical or cultural distributions.
Yeah, and as I think I said to you the other day, I've got a collaborator who works in Singapore who said to me that if people in Singapore are diagnosed with celiac disease, the first question they ask if you're ethnically Chinese is, are you illegitimate? So there are populations where, as far as we know, it's a relatively rare condition. It's generally associated with the more Celtic populations in northwestern Europe.
All right. Okay, we've got a question from Bex. Can the gut heal itself if celiac disease follows a gluten-free diet?
Yes, as far as we know, if you follow a gluten-free diet, the gut should heal completely. The difficulty is always making sure that you're squeaky clean, particularly if you're in a shared living environment. If that odd crumb gets in, then that can undo quite a lot of good. But in theory, if people follow a proper gluten-free diet, which may also involve excluding oats as well as the classic wheat, barley, and rye, then the gut should heal up. It may take 3 to 6 months, so it may not be quick.
Right, okay. Do we have any more comments? No. Oh, David says this is absolutely fascinating, and it is. It is. Right. I think we've got a creative reactions to this. I know you've been working with 3 different artists. Oh wow. Oh wow. This is like scientists reacting to artists reacting to science. Wow. Look at the colours.
Absolutely amazing. Really
yeah, so,
beautiful.
If you want to find out more about this, the description has a link below. And if you want to find out more about how the artist created this work, make sure you stay tuned for the videos at the end of the show. Anyway, thank you very much, Liz. It's really great to see you, and I'm really happy that your kids are really healthy now as well.
And they went to bed to let me do this, which is great. All right, thank you very much.
Thank you, Liz. Right. We're moving on to our next guest now. So we are now talking about poo, or more specifically, how the gut microbiome affects the aging of the immune system. Right, to get to the bottom of this, Dr. Marisa Stebegg, a visiting scientist at the Bagram Institute and from KIMAB, will tell us more about this. Hello, Marisa. Thank you for joining us on the show. And it's over to you. Thank you.
Hi, thanks for the introduction, Andrew. As I hope you can all see on my slides now, I'll spend the next 8 minutes talking about how we grow old on the inside, and also describe a few links between our gut bacteria and aging potentially. I think it's important to talk about aging because it affects all of us. And while we all hope to live long, healthy lives, in reality, we often spend the last years of our life riddled with age-related diseases. As scientists, if we want to try and extend health span, meaning the years of life in good health, we really need to try and better understand how aging affects the different parts of our bodies, so we can try and counteract its negative effects. In my research, I was most interested in trying to understand how aging impacts the immune system, and also in particular the gut. And I'm interested in the gut because it's not just a digestive tube, as shown here. It's actually much more. It's a whole ecosystem containing an estimated 100 trillion microbes, some of which I think are quite nicely illustrated here on this cartoon, which I stole from a book you might have heard of, Gut, which I can definitely recommend. But the gut doesn't only contain these cute microbes. It also harbors the largest collection of immune cells in our body, actually, and also the largest collection of neurons found outside of the brain in a human body. So there's lots going on in the gut. But for this talk, let's focus on our gut microbes. These are not just hungry lodgers who live in our guts and feed on what we eat. They actually help us in many different ways. They help us digest certain foods, for example, and also produce vitamins. They also closely interact with the immune system from an early age, something that I am interested in. They actually help the immune system to mature properly and to help us fight infections and also to prevent autoimmunity. And in addition, microbes can also produce hormones which influence our mood and behavior, giving the term gut feeling a completely new meaning. To add to the complexity, there's also something called the poo print. And this describes the fact that each of us contains a unique mix of different microbes, which are so unique to each of us that they can be used to identify an individual, similar to a fingerprint, hence the name poo print. And it's this unique mix of microbes which determines how good or bad they are for our health ultimately. Unfortunately, in aging, the composition of gut microbes was shown to change, and often not in a good way. This is associated with changes in the composition of certain gut immune cells as well, and a trend for increased low-grade gut inflammation. So in my research, as part of the Linterman lab in Cambridge, I wanted to understand how exactly aging affects gut microbes and the gut immune system, whether we can revert these changes, and what happens if we do. And because it's difficult to study the gut immune system in humans, we decided to use mice instead. For this, we used young adult 2-month-old mice and 2-year-old mice, which in terms of age are thought to be equivalent to an about 80-year-old person. And we started off by just looking at the gut microbial composition in these mice. And this is what's shown here. So these circles, they show the relative contributions of different bacterial orders or families in different colors to the overall gut bacterial mix in adult mice and aged mice. And if you just look at the colors, you can see that the aging has a strong impact on the composition of gut microbes. In adult mice, the red Bacteroidalis are more prevalent, whereas in the aged mice, we saw an increase in the green Firmicutes in this case. What is more, when we quantified bacterial diversity, which means the number of different species which are part of the mix of gut microbes, we saw this striking reduction in bacterial diversity in our aged mice compared to adult controls. And this is interesting because in humans, a similar trend has been described where a reduction in the diversity of gut microbes was actually associated with an increased risk to develop age-associated diseases, which indicates that this drop in diversity could have a negative impact on our health. Question is, can we revert these changes? And to test this, we decided to co-house young and aged mice in the same cage. And we did this because it's known for mice that mice who live in the same cage exchange gut microbes, and they do this by grooming, but also because they actually eat each other's poo. Yeah, you've heard right. And truly, after 2 months of co-housing, we observed that the gut microbial composition in aged mice had normalized. And you can see this here when looking at bacterial diversity again. If you look here at the aged mice before co-housing, you see that their bacterial diversity is again very low. But after co-housing, shown here in light blue, bacterial diversity had increased and was now actually very similar to what we see in the adult mice before and after co-housing. And this indicates that co-housing could serve as a way to rejuvenate the composition of gut microbes in aged mice at least. And this is of particular interest because it was shown for humans as well that people who live together in the same household also share skin and gut microbes between each other, and actually also their pets. So this could indicate that cohabitation in humans could play a role in shaping gut microbes, even though we don't tend to eat each other's poo as mice do. So I found this quite an interesting result, but I was even more excited when I realized that co-housing didn't only seem to affect the gut microbial composition in the aged animals, it also seemed to have an impact on their gut immune system. And that's what's shown here. If you look here, this is a plot that was generated from mice before co-housing, and you can see that there's a reduction in the specific gut immune cell type in the aged animals. And this gut immune cell type I'm talking about is also known as germinal center B cells. And these cells are really important because they give rise to cells that produce gut antibodies. And these antibodies are important to help us to fight gut infections, but they can also bind to the surface of gut microbes and actually shape gut microbial composition as well. So it was quite interesting to see that there was a reduction in the number of these cells in aged animals. What was even more interesting is that after co-housing, the numbers of these cells normalized in the guts of aged animals. And this didn't only happen upon co-housing, we could see the same trend when we performed something called poo transplantation or fecal transplantation, where we took fecal matter from young animals and actively transferred them into the aged animals. So does this mean that by rejuvenating the gut microbes, by transferring gut microbes between young and aged animals, that we can also rejuvenate the gut immune system? Unfortunately, it's still a bit too early to say, but more research will hopefully help us answer this question, in particular in a human setting as well. In conclusion, I think there is very strong evidence for the importance of gut microbes for our health. There are studies that describe a possible impact of gut microbes on aging, but also an impact on an individual's response to cancer treatments, for example, or vaccination. And what's also interesting is that fecal transplants are now actually a common standard treatment in many hospitals for intestinal Clostridium difficile infections. And that will hopefully give us more insights into the long-term impact of fecal transplants in humans. Still, it's very important to remember that this is a hugely complex area of research and there's still much to learn. We still don't really know what a good or bad mix of microbes actually looks like. And we also haven't found out yet how we can best modulate our gut microbes for optimal health in different individuals. I'm sure you all know the saying, trust your gut feeling. But I think it's only now that scientists like me start to realize how true the saying might be. So this is definitely a fascinating space to watch in terms of new scientific discoveries. And with this, I'd like to thank you for your attention and pass the mic back to Andrew.
Thank you very much, Marisa. That, again, really, really fascinating talk about poop. So literally the fountain of youth is poo.
Well, we don't know yet. Maybe not. Maybe a bit.
Right. So let's bring some questions on from our audience. We'll have one from Kate. Does co-housing in mice also suggest the usefulness of therapeutic fecal transplant in humans?
It's always difficult to go from the mouse setting directly to the human setting. I think it's important to do studies in mice just because it allows us to do more experiments that we just can't do in humans. But if something like cohabitation or fecal transplantation actually has a positive impact on humans in terms of aging, that's something we need to look at in humans. And I really hope that maybe the data generated from fecal transplants that are being conducted in hospitals as a treatment for infections could actually give us some data about whether fecal transplants have an impact on aging in humans.
So that was on difficile, wasn't it? Clostridium.
Yeah, exactly. Yeah, Clostridium difficile.
Excellent. Right. We have a question from Praveen Paul. How far are we in the process of identifying gut microbes in humans? Have we identified most? Ooh.
I guess there's two ways of thinking about this. There's the way of identifying microbes by sequencing their genomes. That's something where lots of work is being done, and many bacterial genomes are being built, and we're kind of inferring which kinds of bacteria we carry in our guts from that. The other one is to actually extract the bacteria, trying to culture them, and actually being able to analyze them a bit more. And there is many efforts to increase our knowledge of the gut bacteria that live within us. And we know many more now than we used to. One of the tricky things is that many of them are anaerobic, so they don't grow once you expose them to oxygen. So it's quite tricky to grow them. But yeah, we know many more now than we used to. But I don't think we know all of them yet. That will require more research.
Yeah, fascinating. It's like a whole zoo down there. We have one from Niall. Many aged humans are often treated medically with antibiotics. Presumably, this compromises their gut microbiome, therefore, paradoxically, their health. Yeah.
Yeah, there is a bit of a tricky one. It's definitely the case that if you're an older person and you do take antibiotics, the gut often tends to react in a worse way. And that's actually these Clostridium difficile infections. They occur usually after antibiotics treatment, when the gut microbes are out of balance. And it happens much more often in aged individuals that this infection occurs because the gut is already a bit weakened. And then if you do, add another insult, you make things worse. So yeah, there is definitely a link. And yeah, I mean, we need antibiotics to, protect us from infections that might kill us. But we need to be a bit careful about how we use them because they can have negative impacts on our health.
Yes, yes. Right. Do we have time for one more question? Yes, we do. Verena, are inflammatory diseases like Crohn's or colitis a form of premature aging of gut microbes?
Never thought about it that way. I mean, I don't think so. I'm not an expert on Crohn's disease and colitis, but they are the link between gut microbes and Crohn's is still. I mean, there's definitely a link, but it also seems to be very much an inflammatory disease where immune cells start to overreact and the gut goes haywire. And in that sense, it's different from aging. So there might be certain parallels, but I think they are still very separate in how they affect us, and also probably in the way they occur.
Right. Yeah, thank you very much, Marisa. We're going to check on your artwork from your artist. So do we? Oh, there it is. Wow.
They're all amazing. Yeah. I had 4 artists I was very privileged to work with. Very interesting, amazing artists.
Wow. So there's some like figures there. There's something which looks like a heat map from some kind of microarray. And there's some pictures of mice, I think, in the gut. Yeah, so we've got videos of these, and the artists will show you how they made their work at the end of the show. Right, again, thank you very much, Marisa. I get very fascinating talk about poo. Thank you. Right, now we're moving on to our final talk, moving from fecal to fetal. Have you ever wondered why organ transplants normally get rejected, but the fetus can spend all those months inside a mother's womb without having to worry? So what's happening here? To get to the answer, we will have Anya Arutyunyan. Anna Arutyunyan is a PhD student from the University of Cambridge and the Wellcome Trust Sanger Institute. Welcome, Anna, and welcome for joining us on the show. Right, it's over to you.
Andrew, thank you for having me on the show. It's really exciting to be able to present. Let me know if there's any technical difficulties. Today I'm going to tell you the story about home invasion, or how human pregnancy works. And basically, before I do that, I thought it would be good to kind of give a very, very simplistic recap of what types of immunity humans have. And in particular, here on the left, you can see this diagram that shows that largely the immune cells in our body can be divided into the so-called innate and adaptive immunity. And what this means is that the innate immunity, which is the first line of defense upon any sort of infection or a reaction of our body, is the kind of the initial response of our body to in order to basically fight the infection or any perturbation. And it's usually very non-specific, and it's conducted by the cells that you can see in the circle on the left. And then we have the adaptive immunity, which is basically much more targeted and specific. And it's usually tailored specifically to the pathogen that attacks us. However, this kind of second line of defense is happening much later after the sort of infection or perturbation. And on the intersection of those two, I mean, as with anything in biology, I think there's the lines are very blurry, and evolutionary, it just happens so that the so-called natural killer cells, which will be very important for us in this little talk, they are kind of bridging this innate and adaptive immunity in humans, and they, in certain circumstances, they can perform both roles. And as you can see in these kind of funny illustrations here, these cells basically keep them in mind, and yeah, we'll go to the pregnancy part of this. So first of all, what is a placenta, right? Many people actually kind of are a bit mixed up on this, and placenta is actually a fetal organ that is kind of that emerges in the embryo once it implants itself into the maternal uterus after fertilization. And so at the site of implantation, it develops this kind of thicker layer, and it also surrounds the fetus. And basically, when the pregnancy sort of develops throughout the first trimester, the placenta in this site of implantation grows into these structures that look like these trees. And ultimately, the goal of this is to anchor these tips of the placental villi into the so-called decidua, which is the mucosal lining of the mother's uterus in pregnancy. And what happens after that is these fetal cells, they kind of burst into the decidua, and they travel further, and they sort of, quote unquote, invade maternal uterus. And so the purpose of this is to be able to transform these spiral arteries inside the maternal uterus. And this is done in order to be able to provide an increased blood flow into the space between the uterus and the placental villi, which is a very important thing, because that's where the nutrition happens, basically. The transport of nutrients and gas through this barrier of the surface of the villi and maternal blood. That's how the fetus basically gets nutrients. That's how it regulates its temperature. It gets rid of waste. It also secretes hormones that support the pregnancy in general. And it also, the placenta also serves as a barrier for fighting internal infections. And so you might ask yourself, if this is a fetal organ, then genetically speaking, this is a sort of mosaic thing made up of maternal and paternal DNA, then why is it not rejected? And just to give you some context, I will compare the two scenarios of, let's say, an organ or skin transplant and the embryo implanting in a woman's uterus. So to this day, we don't really fully understand how to sort of match the donors and recipients of organ or parts of organ transplantation, because in most cases, what ends up happening is our adaptive immune system kind of recognizes the foreign cells or the foreign matter as genetically unknown or foreign to the host body. And what happens is there appears a very kind of severe inflammatory response, and this piece of skin or sort of organ can get rejected, and does get rejected in most cases. As you can see here in this diagram, there are many T cells that come and kind of ring the alarm, whereas in the case of placenta and in case of pregnancy, what ends up happening is instead of the adaptive immune cells such as T cells or B cells, this tissue is actually swarmed with the NK cells, which you might remember from my first slide. And what ends up happening is because of the very specific cell type composition of this tissue and the very unique cell-cell communication that happens between the invading cells of the placenta and maternal natural killer cells, the fetus doesn't get rejected. And what does this mean? A bit more in detail. So recently there's been this amazing study that basically profiled the different cell types in this tissue and their networks of communication. And importantly, it discovered that this tissue is basically predominantly populated by the innate by the cells of the innate immunity in humans, which are, in this case, predominantly natural killer cells, maternal natural killer cells, and not as much as the adaptive immune system. So it's kind of a very unique immune environment, exactly for the reason that the communication that happens here is actually kind of between the invading cells of the fetus, which are called extravillous trophoblast, here in purple and also here in purple. And what happens is these invading cells of the placenta interact with the maternal immune cells, which are again predominantly natural killer cells. However, speaking in more molecular biology terms, they do not express the necessary ligands and receptors that would facilitate the communication to the sort of T cells, B cells, and so on. And this is how they manage to stay invisible to the adaptive immune system, and this is quite fascinating. However, communication to the innate immune system happens because there needs to be the regulation of how far this fetus invades and how much it reforms these maternal arteries. And this is of a very high relevance for pregnancy success because basically there are conditions such as preeclampsia, where this invasion of the placental cells doesn't happen enough, and therefore the maternal arteries do not enlarge enough to provide enough nutrition for the fetus to grow normally. And in those cases, the fetus basically ends up being of a low birth weight, which can result in complications up to the fetus basically not surviving. So this is very important to understand. And if there will be a very short take-home message from my little talk, I hope that you will remember that placenta is amazing. It is a fetal organ that is kind of developed by the embryo that establishes the necessary connection to the mother's body to, first of all, provide nutrition for the normal growth of the baby. It also collects waste that the baby produces while it's in the uterus. And it also helps fight internal infections. And one fascinating thing about placenta is that it is made up of kind of chunks of maternal and paternal DNA because it is a fetal tissue, and therefore it is genetically foreign to the mother's body. But it does not get rejected, unlike the sort of tissue and organ transplants, because of the very unique cell type composition of this tissue and the cell-cell communication that happens between these invading cells of the placenta and natural killer cells of the mother, and actually also other types of cells at that very unique immune environment. So I would like to thank a lot of the people who have made this work possible, donors and the funding that was supporting this work. And also, if any of you want to check out the original study, it has been published in Nature with this really cool cover, kind of spearheaded by Teichmann, Moffat, and Hanifa Labs. And also you can check out our lab's website, where we continue to develop these efforts on atlasing reproductive organs in the human body. You can also check out myself on Twitter, and I think we are about to check out the art that was inspired by this talk. Yeah, thank you.
Thank you very much, Anna. Who would have thought the immune system had such an
I
important
know.
role in pregnancy? And I love the name natural killer cells.
It's kind of an unfortunate name in this case.
Yeah. So I know that natural killer cells go around killing cells which are virally infected or unknown cells,
Or cancerous,
non-cell
yeah.
cells, or cancer cells. But do they cause any damage in the placenta at all?
I mean, it's kind of a debate, but largely they do not kill anything in this setting, which is actually quite interesting. I mean, it's quite a unique environment. So it's a very unfortunate name in this case.
It's quite lovely, such an important role they're playing. Right, so do we have some questions from the audience? Oh, yes, from Marco. How does cell-to-cell communication work in pregnancy? Do cells need to be in contact, or do they communicate from a distance?
Right, this is a very interesting question. I mean, at this point, we do know that communication between cells can happen both when they are actually joining the receptors and ligands, kind of being right up against each other. There's also communication through secreted molecules, which means these molecules can diffuse through the media and kind of reach the other cell. So those are the kind of, I'd say, largely the main two types of communication. So it happens both ways, and actually currently a lot of these efforts that I've kind of been talking about, they are translated onto space, so we are able to profile this in space, and it's an open question. I mean, we are researching it at the moment, so yeah, that's kind of the answer. I hope it covers.
Yes.
Yeah.
Right, so we have another question from Jordan. Does the genetic similarity between the mother and the fetus play a significant role in the fetus's ability to invade silently?
Right. So, yeah, basically, the similarity per se doesn't play that much of a role. What does play a role, though, if I just go back here, the only kind of well-known link between genetics and how well this invasion happens is the sort of only well-known link is these two kind of interacting ligands and receptors. So basically, depending on, thank you for bringing up my slides. Yeah. So basically, the fetal extravillous trophoblasts, the invading cells, they express the HLA-C ligand that in its turn interacts with these key receptors on the maternal natural killer cells. And basically, the genetic combinations of certain alleles and genetic groups of these can determine whether the fetus sort of invades very vigorously or it under-invades. And you can kind of end up with scenarios where the fetus actually overgrows and is too big, or it undergrows and therefore it has low birth weight. So yeah, in terms of similarities between sort of mother and fetus, not much is known, but there is this link. And in fact, in African populations, just because due to other genetic sort of pressures that they have to deal with, like malaria and things like that, there's sort of a genetic trade-off. And in African populations, the frequency of preeclampsia and sort of diseases where the invasion kind of happens a bit less than it should are actually more frequent. So you would have a lot of these preeclamptic cases there. Hope that makes sense.
Yes, thank you. Right, we have one last question, I think, from Peter. What level of clinical tests can be carried out to establish the fetus's immune system effectiveness, and how do these tests distinguish them between the mother's immune system?
Right. I think it's a bit challenging to say, and I'm not the best person on, I'm not an expert on sort of clinical tests in pregnancy, actually. However, I would speculate that you could differentiate between the fetus and mother's cells, just genotyping them. That's quite easy. And in terms of the effectiveness of the immune system, I'd say you would need to healthy pregnant woman, first of all. But I think that can be done. And then if you do actually manage to separate immune cells from fetus or mother, then you could, I don't know, possibly play with them in the lab. But again, sorry for this kind of sketchy answer. I'm not the best kind of person to ask. I hope
I think blood tests would be possible. But again, like you said, you need a good reason. Right, so thank you very much, Anna. We're going to quickly go on to your artist's work. Let's see what we've got. Oh, you've had 3 artists. Oh, that's a funny one.
Yes. I was privileged to work with 3 artists, yeah.
Yeah, right. It's a very maternal theme, which is really, really lovely and warm. Again, so if anyone wants to know more about these artists' work, you can watch the videos which are coming up very soon, and also go to the website, and the link is below. Right. Thank you very much.
Thank you.
And it's been really lovely to have you. And thank you very much for watching, all of you at home. This concludes our round of science talks. And also thank you very much for the speakers for giving up their time to tell us all the lovely research they're doing. If you have any further questions, please continue to post them in the comment section below, and we will endeavour to answer them. And make sure you hit that like button. Okay, to play us out, we've got some of the videos of the amazing artwork from Creative Reactions. You can view the exhibition in the link below, and pieces are available for sale. So until next time, stay alert and stay safe. Bye.
Hi, I'm Amy. I'm an illustrator and animator, and I'm going to talk you through the print I've made in response to Elizabeth's research for Pint of Science. When Elizabeth first talked us through her research into celiac disease, one of the things that stuck out to me was the way that she described each person's unique set of T cells to being a bit like the human body's equivalent of a barcode. So I took that barcode imagery and I started inking some black and white loosey-goosey intestines, and then I scanned that image into my computer, and as I did that I wobbled the paper around a bit to create a glitchy effect, and then I edited the imagery, played around with some colours and stuff, and then when I was happy with it I sent it away for risograph printing. So this is my finished print. I chose to use red and blue inks as those colours cropped up quite a lot in the way Elizabeth presented her data, and I've also slightly overset the 2 ink layers as well to reflect the Venn diagrams that she used quite a lot too. So there we go, hope you like it. Bye.
Hello, my name is Elena Arevalo Melville. I am a children's books author. I am both writer and illustrator, an artist, a potter, a map maker. I really like doing Pint of Science because you don't really know what you're going to get. You put yourself forward for it, and then it's on the hands of the gods what sort of research is going to land on you. This year, obviously, we're all stuck at home, and it's going to be a little bit different than previous times. So we're paired up with loads of artists per scientist, and I am really curious to know what my fellow artists are going to do with Marisa's research. Marisa's research works with immunology, the gut, and aging, and how these 3 things combine. And because it has been a very short project, we had to tackle it pretty much like a reaction, like a creative gut feeling in a way. I made 2 bits of artwork. One is a little bit more graphic, and it's based on the concept of the poo print, how we each have a very unique combination of microbes living in our gut, and how we can actually be identified through our gut bacteria. And the other one is a little bit more narrative, and it's about these 2 mice, and one is an old mice and the other one is a young one, and they're both benefiting from being together by having a more diverse sort of bacteria around them that protect them from outside threats. I hope you enjoy them, I hope you like them, and yeah, it's been really exciting to be part of this again. Bye!
Hello, I'm John Hodgson, and this is my latest work called And Does Luncheon a Great Big Mess? It's about diversity and the loss of diversity and the possibility of restoring diversity. I wanted to show visually what diversity looks like. I tried flowers and glowing coal feces and geometric farms and skyscrapers and shockwaves, but settled on these squares with grey to portray uniformity and reds and oranges to show the fire of diversity. This could be about the rainforest, or the Coca-Colanization of culture, or the loss of word power in the meme age when all fashions are, of necessity, fleeting and derivative. But as with so much in this pre-post-COVID era, it starts with immunity and mouse droppings.
Hi, this is Dr. Kate Grant. I've always been fascinated by histopathology, and now I love to recreate microscopic windows on the world of our bodies using resin, and I'm inspired by the immunofluorescent staining colours as well. In response to Dr. Liz Souilleux's work on celiac disease, I've recreated a healthy small intestine transitioning to bowel affected by celiac disease. Underlying the villi are Peyer's patches, which contain immune cells like T and B cells, and these sample material in the small intestine, and they play a role in determining which substances require an immune response. They probably don't function well in celiac disease. I'd like to say thank you to Dr. Souilleux and the 2020 Pint of Science and Creative Reaction team in Cambridge, UK.
I was intrigued to learn from Dr. Marisa Stebegg that our gut health deteriorates as we age, and how housing elderly mice with young mice actually rejuvenates their gut health. She mentioned how the gut biodomes of the elderly in care homes grow to become similar. So I wanted to show what could potentially happen when the young and the old live together, how the young could help to rejuvenate the elderly's gut health. I painted the figures, young and old, very close together, overlapping even, with lots of textures to show all the biochemical reactions in our bodies. And I added sprinklings of gold leaf on the gut area to show the guts reacting from the intermingling. I'm Susan Apps, and these are my creative reactions, Rejuvenation 1 and 2.
Coexistence is the title of my illustration for this year's Pint of Science. I worked with traditional methods initially, drawing, painting, printmaking, and some photography, extracting from Anna's research some of the symbols that I felt were the sort of most important or pertinent, including the fetus, the placenta, DNA, and the immune cells. I took my drawings onto Photoshop, which is a great platform for being able to play and experiment. So I was able to invert some of the drawings, layer them up and make a more complex image. I also introduced symbols of communication, like a little phone that you can see on there, and Morse code, which I'm really trying to play and represent the very fine balance of communication that goes on between the mother's tissue and cells and the baby's tissues and cells in the first trimester, which in turn leads to either the baby thriving or, in some cases, the reverse happening.
Hello, my name is Lelesa. I'm a Spanish visual artist based in Cambridge, where I am also working as a creative director of a UX research and innovation lab. This is my 4th year participating in Creative Reactions, and it's always very exciting to come up with an idea for the research of a scientist. And in this case, Anna's research sparked in me a little bit of humor. My piece is like what it will be the moment that these fetal cells arrive to the decidua. How do they have to behave to not being rejected by the women's body, and accomplish the task of create a newborn. So I was very interested on the idea that they are invisible, but they have to still communicate. So this is my piece. It's kind of like this invigilator or cell from the mother, and the natural killer is kind of like handing out some flyers of how to behave when you arrive there. A little bit of like regulations and rules, and these two fetal cells, one is reading it, is like, oh, we need to make sure that we la la la, and the other one is just, well, let's add normal. I also use one of the slides from Anna's talk as a reference for the creation of the characters. So the colors in the figures of the natural killer cell and also the fetal cells try to replicate the slide of Anna. I don't know if that is a general visualization of these kind of cells, but that's what gives me a little bit of idea. And yeah, hope that you have enjoyed it and you like it, and keep learning science in this wonderful week. Keep safe, stay safe.
Hello, my name is Janelle Harvey, and my contribution to this year's Pint of Science Creative Reactions collaboration is based upon the work by Dr. Marisa Stebegg. Influenced by the iconic designs of William Morris, I have created 3 repeating patterns. This one features a stomach motif on a background of floating bacteria bound by Peyer's patches found in the intestine where the immune response starts. For this pattern, I feature representations of the old and young mice from her research, young mice in white, old mice in pink, on a background of floating bacteria, together with chrono plots taken directly from her research. For this repeating pattern, I have brought together the stomach motif together with the mice motif and representations of the cholera toxin used to initiate the immune response.
My name is Adam. I will be your quiz master for this evening. I certainly hope you're going to enjoy yourself. Here we go.